Look at this tree. It stands perhaps a foot tall, yet it possesses the gnarled trunk, the sweeping branches, and the profound age of a forest giant. You might see art, but we see a stunning display of biological manipulation. This is not magic. This is science. Today, we're not just styling trees. We're mastering the botany, physiology, and molecular genetics that allow a 3-story specimen to thrive in a teacup. Welcome to the Biology Behind Bonsai Trees, the ultimate tutorial on the science of miniature forests. The Biology Behind Bonsai Trees. Section 1. The Principle of Controlled Growth. Bonsai, literally translating to planted in a tray, is an art form rooted entirely in the manipulation of a tree's natural physiological processes. The misconception is that bonsai are a special, genetically dwarfed species. They are not. Any tree species can, theoretically, be turned into a bonsai. The key lies in understanding how plants respond to stress and wounding. The entire practice is an ongoing dialogue with the tree's innate survival mechanisms. We apply stress to redirect growth, not to halt it. Every technique we use, pruning, wiring, repotting, is designed to systematically control 2 fundamental biological systems: the shoot system, above ground, and the root system, below ground. By limiting the capacity of the roots, we intrinsically limit the potential of the shoots, maintaining a state of continuous yet controlled miniaturization. Section 2. Mastering the meristem and apical dominance. The first and most critical concept in shoot development is the meristem. These are regions of undifferentiated cells where growth occurs. They are the plant's engine room. You have 2 primary types we manipulate: the apical meristem at the tip of the shoots, responsible for vertical elongation, and the lateral meristems, the cambium layer that increases girth, and the axillary buds along the trunk and branches. The phenomenon we exploit daily is apical dominance. The tip of the main trunk and branches produces a powerful plant hormone called auxin. Auxin's primary job is to promote upward growth, but simultaneously inhibit the growth of the side buds, the lateral buds, located further down the branch. This is why a forest tree naturally grows tall and conical, because the strongest auxin flow is at the very top, suppressing lower growth. When you perform pruning, you are physically removing the source of the highest concentration of auxin. This chemical removal immediately shifts the crucial Auxin-Cytokinin ratio. Cytokinins are hormones produced primarily in the roots, and their function is to promote lateral bud growth. By suddenly reducing auxin concentration in the pruned area, we allow the cytokinins, which are constantly flowing up from the roots, to take dominance. This shift stimulates the dormant lateral buds to sprout. This process is how we force a tree to become dense, bushy, and dramatically shorten the internodal distance, the space between 2 sets of leaves or branches, achieving that characteristic compact miniature appearance. The constant removal of terminal growth is a perpetual chemical reset button for the tree. Section 3. The root system. Pruning for fibrous density. A healthy forest tree develops a large taproot, a single massive root that drives deep for stability and water. A taproot is biologically inefficient in a shallow pot because it consumes too much energy and space and quickly becomes pot-bound, constricting the soil and hindering nutrient absorption. When we perform root pruning during repotting, we are systematically severing that large primary taproot and the large woody transport roots. This severe wounding forces a response in the plant. Much like with the shoots, the tree redirects its growth energy to produce a dense network of fine, hair-like fibrous roots. This is a defense mechanism. The tree needs to quickly re-establish its ability to absorb water and nutrients to survive. Why are fibrous roots biologically superior for bonsai? They have a significantly higher surface area to volume ratio. This maximized surface area allows for far more efficient absorption of water and mineral ions like nitrogen, phosphorus, and potassium within the tiny soil volume. The large woody roots are for transport and stability. The fine fibrous roots are for feeding. By promoting the fine roots, we ensure the tree gets the most out of its limited environment. The aesthetic result, the beautiful spreading Nebari or flare of the roots at the surface, is a secondary benefit of this necessary biological control. By regularly reducing the overall root mass, we directly limit the size the canopy can grow, maintaining the miniature scale and ensuring the root-to-shoot ratio remains sustainable. Section 4. The substrate, a focus on gas exchange. The soil in a bonsai pot is not dirt. It is a highly engineered substrate designed to satisfy two mutually exclusive demands: water retention and, most critically, gas exchange, aeration. Standard organic potting soil compacts quickly, limiting the oxygen available to the roots. Anaerobic conditions, lack of oxygen, are the #1 killer of bonsai, leading to root rot. Our specialized substrates, typically comprising fired clay like Atacama, volcanic rock, pumice, lava rock, and sometimes organic material like composted bark, work because they are highly porous and geometrically stable. Porous structure. The particles themselves are filled with tiny internal voids that retain water and mineral ions that are accessible to the roots. Macroporosity. The spaces between the particles, the macropores, are large enough to rapidly drain excess water and, crucially, allow fresh air, oxygen, to diffuse down to the roots via capillary action. Gas exchange. Roots are living cells. They perform cellular respiration and require oxygen to convert stored sugars into energy, or ATP, for growth and maintenance. Without this constant supply of oxygen, the roots die and the tree fails. The substrate is thus a life support system, optimized for gas exchange, often lasting only 2 to 5 years before organic breakdown necessitates repotting. The fact that the plant must constantly force energy into new root growth sends a signal to the top that limited shoot growth is required. Section 5. Wiring and secondary growth, the cambium. Wiring is the artistic technique that dictates branch structure, but the biology behind it is secondary growth. The tree's trunk and branches increase in girth via the vascular cambium, a thin cylindrical meristem located just beneath the bark. This cambium layer produces xylem, water transport, cells to the inside, and phloem, sugar transport, cells to the outside. This is how a tree strengthens its structure. When we wrap a wire around a branch and bend it, we are applying external mechanical stress, specifically tensile stress on the outside of the curve and compressive stress on the inside of the curve. The tree, in response, activates its repair and reinforcement mechanisms. The cambium layer on the outer curve of the bend, the area of tension, will produce more lignified xylem cells faster than the inner curve. This differential growth is essentially the tree building a kind of internal splint to counteract the pressure and lock the branch into the new position. If the wire is left on too long, this differential growth will cause the new bark and vascular tissue to grow around and embed the wire, resulting in the characteristic scars or wire marks. The timing of wire removal is therefore crucial, long enough to allow the new lignified cells to set the branch's position, but not so long that the wire completely cuts into the cambium layer, which can sever the vital phloem tissue and kill the branch by blocking nutrient flow.
Section 6. Leaf reduction and photosynthetic efficiency. A small tree must have small leaves. If a tree maintained its full-size leaves, the compact canopy would block light from the interior, leading to the death of inner branches. Etiliation. We achieve leaf size reduction through a technique called defoliation, or tsumikiri. When you remove most of the large mature leaves, but leave the apical buds, you force the tree to spend stored energy, carbohydrates, creating a completely new set of leaves within a very short period. This rapid regrowth is an intense stressor. The tree, realizing its limited root energy reserves and the need to quickly restore its photosynthesis capacity, produces new leaves that are structurally smaller and often thicker. This is a brilliant survival mechanism. Smaller leaves require less water loss via transpiration and are positioned to maximize light capture across the compact canopy, increasing the overall photosynthetic efficiency of the now miniature structure. This technique is a balancing act. It can only be done on extremely healthy, vigorous trees, as the process temporarily depletes the stored carbohydrates, sugars, the tree needs for basic survival in winter dormancy. It is the most dramatic example of forcing the tree to prioritize survival over size. Section 7. Advanced Hormonal Control. Grafting and Bud Selection. The mastery of bonsai extends to advanced manipulation of plant genetics and hormones. Techniques like grafting allow us to introduce different biological traits onto a single tree. For instance, a desirable but weak scion, the new branch, can be grafted onto a vigorous, robust rootstock, leveraging the rootstock's superior cytokinin production to drive powerful growth into the weak variety. This bypasses genetic limitations of the top growth. In development, meticulous bud selection is another form of hormonal control. By routinely removing the strongest, most vigorous buds, which are the highest auxin producers, and leaving the weaker, smaller, and more inwardly growing buds, the artist is perpetually managing the flow of growth hormones, ensuring energy is distributed evenly across the canopy to maintain a balanced, ramified structure. This prevents lanky growth and promotes the delicate branching structure we prize. This constant selective pruning prevents the natural phenomenon of dieback in the lower canopy due to shading and auxin suppression. Section 8. Symbiotic Relationships and Long-Term Health. No discussion of bonsai biology is complete without acknowledging the invisible partners in the soil. A healthy bonsai substrate is teeming with microbial life, most importantly, mycorrhizal fungi. Mycorrhizae, these fungi form a symbiotic relationship with the tree roots. The fungus extends its microscopic filaments, the hyphae, far beyond what the tree roots can reach, acting as an ultra-efficient nutrient collection system. In return, the tree, via photosynthesis, provides the fungus with life-sustaining sugars, carbohydrates. The benefit: in a restricted pot volume, this symbiotic relationship is critically important. It vastly improves the tree's uptake of otherwise immobile nutrients like phosphorus, and acts as a barrier against root pathogens. Maintaining this fungal network is a key reason to avoid overwatering, which can suffocate the fungi, and to use slow-release, balanced fertilizers that don't harm the delicate ecosystem. A healthy bonsai is a miniature, complex ecosystem in itself. Conclusion and final thoughts. Bonsai is often seen as a battle against nature, a constant struggle to keep a tree small. But biologically, it is a delicate partnership. We aren't fighting the tree, we are speaking its language. We use auxin and cytokinin to control shape. We use secondary growth to set structure. We use photosynthesis and cellular respiration as our fundamental energy metrics. Every cut, every bend, and every repotting is a carefully calibrated instruction that tells the tree, focus your energy here. Survive this stress. This tree is a living testament to the sheer adaptability and resilience of the plant kingdom. By understanding the biology, you move beyond mere technique and become a co-creator, sculpting life itself. Thank you for joining us on this deep dive into the true science of miniature forests. Now go forth and grow. And you can fade out with an inspirational shot of the bonsai.